The Hawaiian Islands are isolated in the mid-Pacific, and in that isolation there's been a rapid evolution of many plant and animal species. Species such as the present-day picture-wing flies, descendants of probably a single fly that arrived on one of the islands a few millions of years ago. Honey creepers, such as the apapani, have had a similar history, having descended from a single immigrant ancestor. These, and other animals and plants that you've already seen in the course, are just a minute sample of the diversity of living species in Hawaii. And of course, Hawaii is itself only a microcosm, with only a fraction of the worldwide organic diversity that lives now and which has lived in the past. Indeed, the living diversity we see today is only a small sample of all the species that have ever lived on Earth. For every living species of today, there are hundreds that have lived and gone extinct in the past, and some of these come to us only through their fossilized remains, if we're lucky enough to find them. Let's briefly recapitulate the geological past. Our ancestors, such as Homo erectus, ranged widely across the plains of Africa and other parts of the Old World about 1 million years ago, towards the end of the Cenozoic era. Beyond that, the Mesozoic era, when reptiles were the dominant vertebrates. That began about 225 million years ago. Beyond that, spanning a further 345 million years, the Paleozoic era, including the Carboniferous period with its lush swamps, and the Devonian period, which saw the first major diversification of fishes and land plants, and so on, back through the Silurian, Ordovician, and eventually to the Cambrian, 500 million years and more ago, when the vertebrates first arose, alongside invertebrates, including some really strange examples like this one from the Burgess Shale. During this 570 million years, life on Earth has been very diverse and has evolved the abundant species we find in the fossil record and the living species we find today. You have all this information summarized for you in your wall chart. You'll be using this a lot during the course. As you'll see, the history of the Earth and life's origins go back much further in time than the mere 570 million years I've just been talking about. Another 4,000 million years. To understand these earliest episodes of the origin and evolution of life, we must turn to the living cell. This is a present-day living animal cell, just one of the inheritors of all that time and evolutionary experiment and natural selection. It may look simple, but it's neither simple in structure nor simple in origin, as you'll see. This is the fertilized egg of an echinoderm and contains the genetic information that, as it divides, will direct its development into a complex, multicellular organism, a sea star. These are living plant cells, and you'll recognize them as such because the box-like cell walls confine the cell and its living, moving cytoplasm that contains these small, round, green objects, the chloroplasts. The living cell, plant or animal, is the basic unit of life, and the cells we've looked at so far, including those of the plant, are eukaryote cells. These cells are part of a green leaf. You can see clearly the features of eukaryote cells if you look at the cells arranged in rows that form the white hairs in this flower. Here are 8 of these cells in a row, and here 4, and here just 1 cell, and within it typical eukaryote features. A true membrane-bound nucleus, this is it here, suspended in strands of cytoplasm. Also in the cytoplasm are these small objects jiggling about. They're the mitochondria, organelles typical of eukaryote cells. And don't forget that the chloroplasts you saw earlier are another characteristic eukaryote feature, but found only in plants. They're only absent here because these cells are specialized. But these much smaller cells are not eukaryote cells. They are prokaryote cells, bacteria. And here they are dividing, though speeded up by time-lapse photography. Prokaryote cells are much simpler than eukaryote cells. First, the genetic information, the DNA, is organized in a simple manner. You can see it here as the irregular light areas within this bacterial cell seen in section. The genetic material is not confined within a discrete nuclear membrane, as it is in eukaryotes, but it's free in the cytoplasm, and there are no mitochondria in the cytoplasm. If the cells are burst, the DNA spews out like strands of fine dark spaghetti. Other prokaryote cells are the blue-green algae. These mobile threads are algal filaments composed of rows of cells. They share prokaryotic features with the bacteria. Each of these cells contains DNA in the central region here. No chloroplasts are present. The photosynthetic pigment is bound only to membranes in the cytoplasm. So a characteristic feature of prokaryote cells, bacterial and blue-green algal, is the absence of membrane-bound organelles. It's now quite certain that prokaryote cells are primitive. They are not degenerate eukaryote cells, but rather are the ancestors of eukaryotes. The transition from this to this was one of the biggest evolutionary steps ever taken, and in the rest of this program, we're going to consider how it might have happened. In the Precambrian, conditions on Earth were very different from those of today and supported different forms of prokaryote life. The atmosphere probably lacked oxygen. This idea is not universally accepted, as recent research findings are providing evidence for disagreement. But were it the case that there was no oxygen, there would have been no ozone shield in the upper atmosphere to absorb harmful ultraviolet wavelengths. The oceans probably contained large quantities of organic molecules, reduced salts, dissolved hydrogen sulfide, and carbon dioxide. Anaerobic metabolism would have been the only option under such conditions. Anaerobic heterotrophic bacteria probably broke down organic molecules to produce ATP, and autotrophs may have obtained energy by trapping light or oxidizing simple inorganic molecules. Now, between 4 and 3,000 million years ago, some photoautotrophs evolved a new system for trapping light energy. Instead of removing electrons from a substance, such as hydrogen sulfide, and oxidizing it to sulfur, they acquired the ability to transfer electrons from water. These were the first blue-green algae, and their oxidation of water affected not only the Earth and its atmosphere, but the whole future path of biological evolution, because oxidation of water produces oxygen.
Blue-green algae are still here, even though now they're relegated to a position of relative inconspicuousness. In fact, you can just see them as a kind of scum on the rocks there. They're still around. If you look carefully down there, you can see that there are bubbles coming up out of the water. Well, the bubbles are just the oxygen. And that's just the oxygen that was responsible. That is, oxygen given off by blue-green algae. It was the oxygen that was responsible for the transition of the entire atmosphere. The atmosphere went from something very anaerobic, no oxygen at all, to the present level of about 20% oxygen, something like 2,000 million years ago, plus or minus 100 million years. And it's just those blue-green algae, that is their ancestors, of course, that led to that transition. So one can imagine that early scene where blue-green algae were giving off this enormous pollutant, that is oxygen, into the atmosphere and poisoning just about everything around them.
As oxygen levels increased, many species retreated to anaerobic environments and are still there. But a few evolved biochemical systems that allowed them to tolerate and eventually use the polluting oxygen.
One of the ways of coping with the increased oxygen back then, and a way I think that was very important in the origin of the complex cells, was by symbiosis. Now, what do we mean? Two bacteria, very different kinds of bacteria, probably got together and ended up using the oxygen. The first of these bacteria simply broke down sugars and in fact couldn't use the oxygen at all. The second took the breakdown products and with oxygen burnt them essentially and derived a lot of energy from that. And the two together made a complex. And this complex, after a long period of time and many, many changes, became a very successful kind of organism. And in fact, it's that kind of organism that is the ancestor to all the higher cells. The things that were once bacteria, after time, became the mitochondria that we find in all of our cells, all the plant cells, all the animal cells, and even all the fungal cells.
But how did this getting together happen? This is a modern eukaryote cell, and this is a mitochondrion in the cytoplasm. How did the ancestors of present-day mitochondria originate from bacteria? Well, it seems plausible that a heterotrophic anaerobic bacterium, which you can think of as a proto-eukaryote, may have looked like this amoeba, engulfing aerobic bacteria which were capable of coping with the toxic polluting oxygen. The engulfed bacteria must have continued to live and feed inside their host, perhaps looking like this. This rectangular object in section is a bacterium, and this is its cell wall, all contained in a vacuole that fits snugly around it, though there is a space here. And here there are several clustering around that circular object. But how did we get this picture? Is this thin section a fossil of such a proto-eukaryote? No. Just as it might have happened 2,000 million years ago, a similar event has happened in a living amoeba to provide us with what might be an analogy for a missing link between simple and complex cells. What you're looking at is an electron micrograph of this amoeba called Pelomyxa. It's the white, slow-moving blob in this drop of water. Pelomyxa is an undoubted eukaryote, as I can now reveal that this large circular object is indeed a true nucleus in section, complete with nuclear envelope. But look hard as you can, and you'll not see the characteristic features of mitochondria. These bacteria are a kind of substitute, apparently metabolizing lactic acid, a potentially harmful end product that the amoeba produces by anaerobic respiration. So, both partners appear to benefit by being together. It would seem that they have evolved or co-evolved together, and it seems that mitochondria may have evolved in a similar way. It would seem that the outermost membrane originated from the vacuole of the original cell that engulfed it, and that the now much folded inner mitochondrial membrane and its chemical activities derive from the cell membrane of the engulfed ancestral bacterium. There are other living organisms which also give us clues as to how these other eukaryote organelles, the chloroplasts, may have arisen by a similar co-evolutionary symbiotic event. Having seen green plant cells, you might think that it's just rather an active one, but it isn't. This is Paramecium bursaria, a single-celled protist that moves by the beating of cilia that clothe its surface. Other species of Paramecium are usually colorless. But this one isn't. It's quite green. Green because it contains a unicellular and once free-living alga called chlorella. The algae are the green blobs embedded in the Paramecium. So you're looking not at one organism, but a sort of committee where some degree of cooperation has evolved between its members. The green alga, by photosynthesis, produces substances useful to the animal cell. The alga apparently can use waste substances produced by the animal cell as a source of mineral nutrients. And there are other modern parallels that indicate how common this kind of symbiosis is. At low tide on the coasts of Brittany, you'll find this beast emerging, a little flatworm called convoluta. They're about 3 millimeters long and green because they contain thousands of single-celled algae. Each flatworm begins life without the algae, the colorless larvae infecting themselves by eating the algae which live freely in the sea. The algae then become a complete life-supporting system as the flatworm stops feeding, and instead uses substances produced by the algae. Similarly, this green hydra contains lots of chlorella. Here you can see a chlorella cell snugly enveloped in a vacuole within a cell of the hydra. In the sea, you'll find a mollusk, a lysia, that in eating seaweed sucks out the seaweed chloroplasts. The chloroplasts enter cells lining the gut, where they produce substances useful to a lysia, as well as providing protective coloration. And I could go on to describe other algae that live in the coloured mantle of the giant killer clam, the algae that live in coral, the symbiosis of fungi and algae that is lichen, all examples of common successful co-evolution. There is now very good evidence that the chloroplasts of green eukaryote cells have been derived in a similar way, and that the chloroplasts we see are the descendants of one-time engulfed blue-green algae. And just as the double membrane of the mitochondrion is of hybrid origin, so it may be that the chloroplast, and this is one from a green plant, that the outer membrane derives from the membrane of the vacuole of the ancestral cell that once engulfed it, and that the inner membrane in yellow, which is much folded, corresponds to the outer membrane of the blue-green alga that was engulfed in a vacuole, and that nothing now remains of the vacuole between these two closely opposed membranes. This is the most plausible explanation for the origin of chloroplasts in the green and red algae and in all higher plants. But compare this chloroplast with this one from a brown alga. What's the difference? Well, the margin of this chloroplast has 4 membranes, not 2. It seems in this case that the membranes picked out in yellow and blue correspond to the usual double envelope of the chloroplasts you've already seen, but the remaining outer membrane picked out in red corresponds to the cell membrane of a eukaryote cell, and the outermost membrane picked out in white to the cell membrane of the ancestral cell that engulfed the eukaryote cell. So it seems that the chloroplasts of the brown algae derived not initially from a prokaryote blue-green alga, but from a green eukaryote alga, a case of parallel evolution. Symbiosis has also been invoked to explain the origin of cilia. It's a coat of cilia that are responsible for propelling paramecium through the water, helping this ciliate graze, or these in darting about. Cilia, and their longer versions, flagella, are universal, but it is thought that they were once free-living organisms rather like these bacteria, spirochetes, that you can see wriggling about, and that cilia were derived from them. Again, there seems to be a modern counterpart of this association. In the gut of this Australian termite lives a protozoan, Mixotricha paradoxa, that helps the termite digest wood. But the cilia that propel Mixotricha with their synchronized beat are not cilia at all, but attached spirochetes. Mixotricha appears to be another one of these symbiotic committees. But the evolutionary contribution of spirochetes may not have stopped there. Every time a eukaryote cell divides, the chromosomes divide and move as 2 equal bundles to the daughter cells, apparently due to the contraction of microtubules. Here you can see microtubules attached to the darkly stained chromosome at the left. The synthesis of microtubules is organized by structures similar to those that control microtubule synthesis in cilia, so perhaps, and it is tentative, symbiotic spirochetes may have given rise to both cilia externally and microtubules internally. But what hard evidence, fossil evidence of known age, do we have of eukaryote and prokaryote cells? This is presently the oldest known eukaryote fossil, dated at 1,500 million years. These are the oldest known fossils of prokaryote cells, some kind of blue-green algae and bacteria, at around 2,000 million years old. And even more ancient, at 3,500 million years, are these stromatolites, which we believe to have been formed by the activities of blue-green algae. And this? Well, this is a man-made pseudo-fossil and represents the latest attempts by Sidney Fox of the University of Miami and others to show that such man-made models of early protocells look very similar to ancient microfossils, of which this is one. This is another of their artificial fossils. And this, a genuine ancient microfossil. So what are these pseudofossils? They're composed of microscopic spheres, microspheres, which have been formed spontaneously in the laboratory under conditions that could have existed on the primitive Earth. If mixtures of amino acids are gently heated, they link together chemically, and if such a proteinoid material is then mixed with water, these microspheres form spontaneously. They're about 10 microns across. In section, they appear to have a layered membrane. Another surprise is their ability to grow by budding, and that inside them it's been possible to set up simple biochemical transformations. But the heating of the amino acids that produces the proteinoids of these microspheres does not result in a random chain of amino acids, but in a preferred order, determined by the shapes of the amino acid molecules themselves. So it seems possible that the primitive Earth possessed the conditions that enabled simple microspheres to form spontaneously, and that over the next 3,000 million years, protocells emerged, which we now find and recognize as microfossils, and by way of the stages you've seen in this program, these eventually gave rise to modern cells.